So today's we have a very distinguished speaker, uh, Dr. Nilkamal Mahanta from Chemistry Department from Indian Institute of Technology Dharwad, and he will be talking about this year's Nobel Prize in Chemistry, and that is about genetics research. And what I heard, I'm not from a chemistry background, and it's very happy to listen somebody talking about the importance of today's this year's Nobel Prize in Chemistry. And what I read about it, it is one of the very fundamental thing which they have they have invented, that it is so fundamental. It is going to change all gene therapy, it seems. That's what I read. And we'll be listening it from Dr. Nilkamal. And I now request Dr. Partha Pradimdas, my colleague, to introduce Dr. Nilkamal to the audience. So over to you, Partha.
Yes, sir. Thank you. Am I audible?
Yes, yes.
Yeah. Good afternoon, everyone. So it's pleasure. I'm just going to introduce Dr. Nilkamal Mahanta. So it's pleasure welcoming Dr. Nilkamal Mahanta to this webinar in the Department of Physics of NIT Karnataka, Surathkal. So let me read out a few lines from his achievements and CV, etc. So Dr. Nilkamal Mahanta received his bachelor's degree in chemistry from University of Delhi in 2005 and master's in chemistry from IIT Delhi in 2007. After a brief stint of lectureship at Zakir Hussain College of Delhi University, he went to Texas A&M University, Texas in 2009 to pursue higher studies, where he worked on vitamin biosynthesis and enzyme chemistry under supervision of Professor Tag. It's difficult to pronounce the name. So T. Bagley, which earned him a PhD in 2015. Thereafter, he joined Professor Douglas Mitchell's lab at the Institute of Genomic Biology and Department of Chemistry at the University of Illinois at Urbana-Champaign to conduct postdoctoral research on antibiotic ribosomal natural products and peptide protein modifications. In December 2018, he joined the Department of Chemistry at IIT Dharwad as an assistant professor and currently serving as the faculty associate of the department, in addition to other academic and administrative responsibilities of the institute. His research interests are in the field of chemical biology, bioorganic chemistry, enzyme mechanisms, and antibiotic, anti-cancer natural products. Notable recognitions in his academic career include Professor M.K. Rastogi Award and Professor Seshadri Award from University of Delhi, Post-Graduation Merit Scholarship from University Grant Commission India, Merit Cum Mean Scholarship from IIT Delhi, F.A. Cotton Research Travel Award and Dow Chemical Research Scholar Award from Texas A&M University, C.S. Postdoctoral Award from University of Illinois at Urbana-Champaign, and Travel Award from American Chemical Society. With these few words, I welcome Dr. Mahanta for this talk.
Thank you, Professor Partha, for this nice introduction. And I also thank Department of Chemistry, Department of Physics, NIT Surathkal, for giving me this opportunity to talk in this colloquium they have organized on this year's Nobel Prizes. Am I audible to all of you?
Yes.
Okay.
Yes.
Okay. So here today I'm going to discuss about this year's Nobel Prize and how this this technology that they have developed in this recent years has almost transformed medicine and other fields that are really revolutionized the genome editing process. So here in this figure, you can see a protein complex that is used along with RNA to cut the DNA. So here is the genetic surgery that is going on to cut DNA at a particular location. and that can be used for different purposes, including medicine, agriculture, as well as biotechnology. So the title I have given is Genetic Scissors because it cuts, a technology that has revolutionized genome editing. So this is 2020, and the technology was discovered or first reported in 2012. So you can imagine that within just 8 years of its discovery, this technology has gained immense importance in different areas. So I thought I'll just give a quick introduction, or I'll just introduce you to the scientists that own this Nobel Prize, then little bit of background on which they have built up this discovery, and then this particular editing technique is known as CRISPR-Cas9. This is a big term or a kind of a complicated acronym that I'm going to break it down in later slides, and then the applications and future implications of this technique. Okay, so moving on to the next slide. So here are the two scientists that I'm going to talk about here. Their work is Professor Jennifer Doudna. She is a Chancellor's Chair Professor at the Department of Chemistry and Molecular and Cell Biology at the University of California, Berkeley. So this kind of work, this is a highly interdisciplinary work, and you can see that she is associated with two departments that are that work in the realm of chemistry and biology. She is also an investigator of Howard Hughes Medical Institute, which is one of the big private funding agencies in USA who funds mainly the life sciences projects which have huge potential. Originally, she is from Washington, D.C. The next laureate is Professor Emmanuelle Charpentier. So she is from France originally, but at the time of this discovery, she was associated with Sweden, Umeå University, Sweden, and University of Vienna, Austria. But right now, she is the director of Max Planck Institute for Sciences for Pathogens in Berlin, Germany. So this is a sister institute of the Max Planck group of institutes. Okay, so here is the file picture of the two scientists that shows that they are present in a conference here. So interestingly, they did not work together. Probably they did not know each other professionally until 2011. So they published this discovery in 2012, and you can imagine that they collaborated across the ocean. She was in Europe and she is in USA. So a collaboration fostered between them to take out to bring out to this level of science that we are going to discuss today. And historically, this is the first two women scientists that own a Nobel Prize. So this is one of the encouragement for the women scientists as well, that there are not too many women scientists that were awarded Nobel Prize in recent years. So this is one of the glowing example of women in science. And they're also the 6th and 7th women in the history of chemistry Nobel Prizes. But they won several other awards jointly. So there are many other scientists also associated with this discovery. We'll come to that in the later slides. Okay, so this is the award-winning paper, or I will say the landmark contribution that they made in 2012. So it was in 2012 they published in Science. The title of the paper says a programmable dual RNA guided DNA endonuclease in adaptive bacterial immunity. This is a very cryptic title. I'm going to break it down. What does that mean? What does dual RNA mean? What is the endonuclease mean? And what is this adaptive bacterial immunity that they're talking about in this paper? But the thing is that this is called as CRISPR-Cas9. So as I told you, people uses CRISPR. They even don't use Cas9 these days, but it's actually Professor Doudna itself says that without Cas9, CRISPR does not mean anything. So you have to mention both the terms to mention this genome editing technique. So what is CRISPR? So let us move on to the next slide. But before that, I want to build down to the very basic principle of life, or it is known as the central dogma of life, or it is also known as central dogma of molecular biology. So this is this particular year's award is mostly in the biological science, biological chemistry, or molecular biology. So it involves a lot of examples from cells and life. So here you can see that this is the principle of life on which it is based on. The DNA, it is converted to RNA and it is converted to protein. So this is the flow of information from the nucleic acid to protein. So DNA is the gene, or a piece of DNA, you can call it as a gene, and that contain the lot of information that is imparted to the protein. Now protein form a lot of functions in our body. They carry out various functions, mainly hormones, So you can see, or they are messengers. Hormone, I can give you an example of insulin that people that controls the blood sugar in our body. And there are a lot of other hormones that proteins are made up of. Then enzymes are the biocatalysts that carry out a lot of reactions in the body. Then proteins also perform structural roles in our muscles. Proteins are very important. Myosin and actin, those are the two proteins that are important. Transport, so proteins are, a lot of proteins are involved in transports. One major protein is hemoglobin. Without hemoglobin, you cannot carry oxygen in blood. So, and myoglobin, which stores the oxygen in the cells. So these are some of the proteins that is responsible for transport a lot of things inside our body. And another important use is the antibodies, or the defense system of our body. When a virus attacks our body, or any foreign material attacks, living system attacks our body generates, or immune system generates antibodies. Antibodies are nothing but proteins that usually target those foreign material. Now, this DNA, there is a simple base pairing rule, which is known as ATGC pair. So A pairs with T double hydrogen bond. So these are two hydrogen bonds. And G pairs with C by a triple hydrogen bond. So these are three bonds that are present in this pairing rule. And this pairing is conserved in the DNA helix. Now, this conversion of DNA to RNA is known as transcription. These are a little bit jargons in molecular biology. Then RNA to protein is known as translation. And if it goes from RNA to DNA, this is called reverse transcription. This is the basis how viruses attack us, like a COVID-19 or even HIV virus. They use reverse transcription pathway. And DNA can make DNA for itself. This is known as replication. That is all like cloning of itself. So this is replication. So this pathway is powerhouse of Nobel Prizes. Almost all the steps in this pathway is awarded Nobel Prize. In fact, in 2010, our Indian origin scientist, Professor Venki Ramakrishnan, also got the Nobel Prize for discovering the machinery or ribosome that makes protein. So this is a very essential pathway on which this is built. So any problem or any defects in protein can be corrected by correcting the gene. So this is, as I said, the gene is the ultimate thing. If you mutate or if you do any disruptions in the gene, it will be implicated in the protein. So people always try to do genetic engineering to perform a lot of activities. So why genetic engineering is important, and why this pathway gain, or why this discovery gain importance with its discovery. So genetic engineering, or people use gene disruptions or gene targeting to understand the function of a gene. If you mutate a gene or if you silence a gene, you are not going to get its function. People then establish the function of a gene. People use genetic engineering to cure genetic diseases. So there are a lot of genetic diseases which are implicated by a faulty copy of a gene, or there is some disruptions in a gene or mutations in a gene. So that will come to some of those diseases in the later part. Then people use it for agriculture to produce better plant varieties, genetically modified crops. Okay. Then it is extensively used in food industry and biotechnology. So genome editing is not new. People have done this from long time back, but they did not have a tool like CRISPR-Cas9. So that is why I'll show you some other techniques people normally use to edit a genome or target a gene, but CRISPR-Cas9 has changed everything. So I'm going to talk to that. I'm going to come to that discovery right now. Now, what is CRISPR? I told you it is an acronym. It's a big abbreviation. So CRISPR is clustered, regularly interspaced, short palindromic repeats. So it's kind of a big acronym that scientists has given in 2005, I think 2002. I'll come to that. So, but it has been continued after that. And what does that mean? I'll come to in the next slide. And Cas9 is very simple. That is CRISPR associated protein number 9. So there are many Cas proteins. It's up to 1 to 13, I think, 1 to 12, 1 to 13. So, but 9 is the one that these professors or these scientists have used for editing. Now, in short, this technology can give you let you do precise genetic surgery. So you can cut paste a gene. So this is kind of remarkable, because there are thousands of genes and billions of genes in our in our body and in cells. So to precisely cut a particular gene at a precise location is not a trivial work. It needs a lot of precision, and because it may lead to a lot of side effects also if you do it wrongly. So Cas9 protein can cut the gene. So here you can see it has cut, and you can insert the new piece of DNA here to give you the function that you want. So you can edit the gene, cut paste the gene, or you can also silence the gene. So this is precise genetic surgery. So that is the power of this technique that you can do in all different organisms as well as even in humans. So I'll come to that in later slides. So discovery of CRISPR. So CRISPR was not discovered by these scientists. So it was first reported in 1987. So here I'm going to discuss what is actually CRISPR is. So look at this diagram. This is a kind of a complicated diagram you can see. But here I'm talking about this. It says the clustered, regularly interspaced, short palindromic repeats. So here you can see that this pink region, these are clustered. Okay, they are clustered, they are short. Okay, they are short meaning they are only 21 and 40 base pairs. So there are 5 of them here. Okay, they are regularly interspaced. So there is some interspacing sequences in the middle. So you can see that these colorful sequences in the middle, they are not the bacterial stuff. They are something else. So those are the spacers in between these repeats. Then they said palindromic. Palindromic meaning something that reads same from the forward and also from the backward. So like take the word madam, M-A-D-E-M. It will read M-A-D-E-M from the front side. It will also read M-A-D-E-M from the back side. So those kind of words are known as palindrome. In case of DNA, it is actually the basis that is palindromic. For example, take this example, TTA-ZCAC. So these are the nucleotides. It reads one front, and this is from the back side, it reads the same, TTA-ZCAC. So this is a palindromic. So these sequences are palindromic, they are clustered, they are regularly interspaced. That means there are spacers in between in regular positions, and they are also short. So this is the terminology. This is kind of a big acronym that was first discovered in 1987 in bacteria. So it was discovered in E. coli, that was the bacteria. It was also discovered in archaea. Archaea is also one kind of kingdom, microbial kingdom. That is one. It was discovered in 1993. So when I discuss CRISPR, I have to take the name of Professor Francisco Mojica. So he is kind of a father figure of CRISPR. And he was also one of the strong contenders of Nobel Prize, but some of his discoveries were kind of underappreciated, as you can see. So here in 2012, he said that these genetic locus is present in all kinds of bacteria and all kinds of archaea. Now, why that is present in all kinds of bacteria and archaea? That has something has to do with a very vital function that bacteria has to do. Okay. Then in 2005, he coined the term CRISPR. That the name was given by this professor in Spain. Okay, University of Alicante. And then he said that these intermediate regions, as shown here, the colorful regions here, are coming from viruses. Now, what virus DNA has to do with bacterial DNA? That was the landmark discovery that was underappreciated at that time. It was discovered. It was published in a very specific journal, so it was not in Science or Nature. So he was the person who says that the intermediate region, the pink regions are from bacteria, but the other regions are from viruses. So how virus DNA has had incorporated into bacteria, that was his landmark discovery in 2005. And based on that, this current state of CRISPR-Cas9 has come. So the basic science discovery is very important when it leads to technology that becomes even more highlighted. So based on that discovery, the scientists told that it is actually a immune system in bacteria. So what bacteria does, it uses this system to protect itself from viruses. So bacteriophage is a kind of virus that attacks a bacteria. So it's a virus. In nature, all these microorganisms are fighting among each other. They want to eat each other. So Darwin's theory always applies that the survival of the fittest. So one that will be fitter, or that will have a weapon to take care of these foreign organisms, that will survive. So similarly, here is the virus. It is known as bacteriophage. Here are different bacteria the virus actually inject the bacteria with its DNA, and it is going to hijack the bacterial system. It is going to kill it. So how does bacteria protect itself? So that is the discovery they have made. So in this first step, you can see the virus has injected the DNA to this bacteria. So bacteria is dead. The cell wall is broken, and all sorts of disruption has happened. But with the repeated attacks, what bacteria will do, it will cut a portion of the viral DNA and it will incorporate into bacterial DNA. That is why those spacers I was discussing in the previous slide are off from the viral origin, because this red piece is the virus DNA that is in the brown piece. So bacteria has what it has done, it has created a memory chip in its DNA that this virus has attacked me earlier. I have to design a strategy to kill this virus when it attacks the next time. So in the next time, the other protein, when the virus attacks again, because of this signature motif, it will sense that the virus has come again. So it will activate the Cas9 protein, and it will cut the virus DNA. So once the virus DNA is cut, it is dead. So it is kind of a defense system of bacteria. So let us a little bit understand in terms of biology also. So this is similar to vaccines that we do. So in case of vaccination, we are injected with a dead virus or attenuated virus. So the virus, our body produce antibodies against that virus. So it is kind of giving a training exercise to our body that when this virus, the live virus will attack us next time, you are ready. The antibodies are ready to kill that virus. So it's kind of a vaccination strategy of bacteria that is present in nature. So here is kind of little bit biology. So first step is known as the adaptation or the creating a memory. So here the virus has injected the DNA, the Cas9 protein of bacteria. Here is the Cas9 protein. So Cas9 protein will cut the DNA, and those pieces will be incorporated into the spacer. So that's where the viral origin is that. So what it has done, it has created a memory. In the next step, when the second attack by the virus, it uses the memory for defense. So when it attacks, when it inserts, the virus actually inserts its genetic material, then the bacteria will sense it. So what it will do, it will activate this CRISPR-Cas system. It is otherwise silent, for example. So what it will do, that DNA will be converted to RNA. As I told you, the conversion of DNA to RNA is known as transcription. This is kind of a term that is used. Okay, transcription. So it produces the CRISPR RNA. So that is the CRISPR RNA. This CRISPR RNA will form a complex with the Cas9 protein, and that Cas9 protein will cut the the viral DNA. So this step is known as interference. So adaptation, that means creating a memory. Interference, that is using the memory to kill the virus. So this kind of a defense system of bacteria. Now virus also, this is a topic of another talk maybe. The virus also found another way to tackle this CRISPR-Cas system. So Darwin's theory is everywhere. So virus know now that bacteria have developed a strategy. Now it has to become smarter. So virus now design some other strategy to take care of this CRISPR. But we'll talk about that in different talks. Okay. Now, what is the discovery that is made by Charpentier? So this is Professor Charpentier. She was studying a protein or the simple system CRISPR-Cas9 in Streptococcus pyogenes. So Streptococcus pyogenes is kind of a organism that is a pathogenic organism. It does scarlet fever. It is kind of a disease-causing organism. It has a simple CRISPR-Cas9 system. At that time, she was associated with Umea University and University of Vienna. Okay. Now, she said that it requires a trans-activating enzyme, trans-activating CRISPR RNA, and trans-activating CRISPR RNA, and it uses a RNase protein for full maturation of the CRISPR system. So it is a little bit complicated. Let me go step by step. So in the first step, this is the full operon of bacteria or genetic locus. Here is the CRISPR-Cas9 system. Here is the Cas9 protein. Here is the CRISPR system. Okay. Then there is she said that there is something else. There is something else that is required for this full function of this immunity. This is known as tracer RNA. It's kind of a non-coding RNA. That means it does not make protein. Non-coding RNAs are RNAs which do not make protein. So once the virus attacks this bacteria, the transcription will happen. So this is the pre-CRISPR system. Then it uses a protein known as RNase 3, or RNase 3 is a protein that cut RNA. So it will cut the RNA, and this tracer RNA will form a complex with that. okay, with that particular CRISPR RNA, and then both of them will form a complex with the protein Cas9. So you have to understand that here there are two systems. One is RNA, one is protein. Protein is the Cas9, and RNA is dual RNA. That's why the title of the talk was dual RNA. One is CRISPR RNA, one is tracer RNA. So these are the two RNAs that are required for the bacterial system to bind the viral DNA and cut it. So here it has cut into two pieces. So this is the immune system of bacteria that she has discovered. So the landmark discovery or seminal discovery by Professor Charpentier is that she discovered that dual RNA is required for full activation of this immune system. So this is CRISPR-Cas9 type 2. This is the simplest one. That is what these two professors have designed, and they have programmed it to use for general applicability. So now in 2011, so she did this discovery, and she was a microbiologist. She's not an RNA expert. She thought that I should collaborate with somebody who is very expert on RNA biology. So what she did, actually, fortunately, in 2011, they were both introduced in a conference in Puerto Rico. Professor Doudna and Professor Charpentier, they became friends in that conference, and they started to collaborate on this topic. So it was a collaboration across the ocean. So she was in Europe, she was in USA, and they both started to collaborate on this interesting project, that how this CRISPR-Cas9 thing work, how it imparts immunity, and how it can be generalized for general applicability and genome editing. So what these two scientists have done is they have found a way to study this protein better. So they studied the Cas9 protein. So here, the blue protein here is the Cas9, and this is the CRISPR RNA that has the complementary sequence to the foreign DNA. So you can imagine this, the viral DNA in case of bacteria. So the CRISPR RNA will have a bonding, or you can have an interaction with the viral DNA. Then the CRISPR RNA will have another interaction with the tracer RNA. So it was discovered by the Charpentier group that Professor Doudna has shown in their paper in 2012, that you can put them together, they will form complex immediately. They will cut the DNA in the two positions. Now, in the next step, they thought that why don't they make the system simpler? So what they did, they joined these two RNAs. So 3' end of one RNA is joined to the 5' end of the another RNA by a linker. And this linker has nothing to do with the activity of the protein. It does not interfere. So that was the beauty of the system, that they could design a custom-made RNA to target any DNA. So it was taken from a bacterial immune system, but it was made for general use. So in the next step, they have shown that if you use this designer DNA, or they call this a chimera because they have joined the two RNA positions, it will complex with the protein, and then it will bind to the foreign DNA, and it will cut that into two pieces. And that is the beauty of this system. So they have also studied this Cas9 protein. They have shown that it also need a motif known as PAM. PAM is known as protospacer adjacent motif. I have shown in the previous slide. In case of this Streptococcus pyogenes, it is known as NGZ motif. Okay, and they have also shown that the nuclease has two domains. One is RUVC. This kind of a nuclease is a protein that cut DNA. I'm going to show the reaction that it does. And then HNS is another domain, so it will cut in these two positions. So when it binds, it is kind of an unzipping of DNA. So DNA is a double helix. It just you can think of unzipping of the chain. Okay, so it will unzip the DNA, the RNA will bind, and then the Cas9 protein will cut the two strands. So this is the targeted strand because the RNA is binding. This is the non-targeted strand. Okay, these are the two strands of DNA. So this is now customized. People started making synthetic DNAs, synthetic RNAs to target any DNA. So previously it was bacteria has designed this system to target viral DNA, but now Professor Doudna and Charpentier has designed this RNA system to target any DNA. You can cut in any cells, human cells, eukaryotic, prokaryotic cells, to do any kind of any DNA of interest. You can target, you can cut at this particular position where there is a PAM motif, and this particular DNA is known as guide RNA, because it guides the protein to cut the DNA. So it is kind of a guiding system. So this guide RNA is now commercially available. You can give the DNA sequence, people will make the guide RNA. So it is kind of commercialized. It has revolutionized the editing process. Now at this until now I have shown that it cut the DNA. What happened after cutting the DNA? So what you are going to do that? So this gene editing uses the DNA repair pathways of the cell. So when our DNA or our cells are always in constant like UV radiation or any kind of radiation damage or reactive oxygen species, those can cut the DNA also. They can damage the DNA also. So cell has a DNA repair pathway. Otherwise, people will end up with a lot of cancers all the time, because the mutations has to be corrected. That is given by nature. So the DNA repair pathways are also given Nobel Prize in a couple of years ago, 2014, I think. So DNA repair pathway are kind of enzyme system that can join the DNA when there was a cut. For example, here you have cut the DNA. So then one system is known as non-homologous end joining. So here it will try to join the DNA, but this process is error prone. It will lead to deletion or insertion of bases, and it will lead to the inactivation of the gene. So you can inactivate or you can target a gene, you can silence a gene by cutting the DNA, because after cutting the DNA, the non-homologous end joining, which is a error prone process, it is not going to repair it fully well. So there will be mutations always, and it is going to kill the gene. Okay. Otherwise, you can edit the DNA. After cutting, you can provide a sequence repair template. It is known as the repair template will provide the base or what you want to put in there. So you want to edit the piece of DNA where you want to where you have made the cut, and you can come up with a new DNA. So this is known as gene editing. So this technology you can use for gene silencing or inactivation of a gene, or gene editing, or you can put the gene of interest that you want. For example, a disease that is caused by genetic disorder, you can cut the piece of DNA that is defunct, and you can put the fresh copy of the gene which is functional. So you can cure the genetic disease in that way. So that is why this technique is becoming popular. So as I said, this is a customized tool now. So what you need, two things. One is this is called single guide RNA, because it is the mixture of the two RNA system. I told you, CRISPR RNA and tracer RNA. Tracer RNA was discovered by Charpentier, and CRISPR RNA and tracer RNA joining was discovered by both of them. And they showed that when you make a complex with the Cas9 protein, it will cut the DNA. And when you cut the DNA, it will be repaired, but there will be deletion, obviously. So the function is gone, so it will be silenced, or you can insert your piece of DNA, what you want. So editing and silencing, you can do whatever you want. In any gene, any genome, human genome, most complex genome, you can do this kind of surgery and repair. Okay, now this is the surgeon, or I'll say the protein or genetic surgeon, because it is the protein that makes the cut. The RNA will only give you the direction or the position where to bind. It is the protein that is going to cut. So here the crystal structure was solved in 2014. It was published in Cell. You can see the blue portion here, or blue domain here, binds the RNA. The cyan and the gray are the two cut sites. So here the cut domains, you can cut here, and the orange domain here is the palm interacting domain, because there is a motif here that is important for the Cas9 to bind the foreign DNA. So one of the limitations also is there, the palm motif has to be there for the Cas9 to bind, but the motif is very simple. It is NZZ. So N meaning any nucleotide, Z meaning guanine, the another Z meaning guanine. Only 3 nucleotides. So there are anywhere you will find that kind of motif. So it is kind of easier to target that motif. Now, when I tell it is a nucleus or it is cut the DNA, where it is acting in terms of chemistry. In terms of chemistry, it is breaking this bond. So DNA, the backbone of DNA is a phosphodiester bond between the two sugar moieties. The phosphodiester bond joins the two bases, and this bond is hydrolyzed. Actually, this is a hydrolysis reaction. You break it and you break the phosphorus oxygen bond. So that is what the Cas9 nucleus does. One on the target strand, one on the non-target strand. So this is kind of a beauty of this system. It is spontaneous. As soon as you put all this thing together, it will go and do the job. So in practice, how people do it, in lab, how people do it. So what they do usually, they put the Cas9 protein, they put the guide RNA, they will put it, both of them in a plasmid. So plasmid is nothing but a circular DNA. This is kind of a nomenclature in molecular biology or biological chemistry. So the plasmid, then you transfect into cell. Transfect meaning you put it inside the cell, that is transfection. So then once it goes inside the cell, uh, the um, the DNA will be unzipped, the guide RNA will bind the target strand, and the non-target strand will, and both of them will be cut by the nucleus. And after that, the repair process will happen, which is error prone and which is going to make to gene deletion. Okay, now let me go through the timeline little bit, because this discovery is very important, although this discovery was 20 years old. So in 1987, it was the first report of the CRISPR that there is a motif, there is a group of DNA which are grouped together, but nobody knew the function. It was in 2000, the CRISPR term was coined by Francisco Mujica. It was in 2000, actually no, it was in 2002, it was termed by Francisco Mujica that the CRISPR means this is the foreign DNA. Then it is the 2005, he showed that the CRISPR locus is ubiquitous and the foreign pieces are from virus. That's where the immunity function came into play, that bacteria uses this kind of system to target viruses. So in 2007, the first experimental evidence of virus, the bacteria killing the virus using this system came into play. So this was also one of the landmark in this discovery. Then some more, in 2008, the CRISPR attacks the DNA target. In 2009, some other CRISPR system. There are 3 systems, 1, 2, and 3. These scientists have used the most simplest of all, that is number 2 system. So number 2 system is actually customized, and people are working on other systems also. Then 2011 discovery by Charpentier that you need a tracer RNA and the guide RNA to make this. Then 2012, it was the science paper that won the Nobel Prize, that you can join the two RNAs to target your DNA. Then 2013, it was shown that you can edit human DNA. So this was also one of the science paper that was discovered. That was also, and he was also the contender. I'll come to his. And 2014, some more discoveries. Now after that, there are a lot of genetic engineering has taken place to the protein itself. A lot of systems has been developed, and it has been now in clinical trials also. Okay, so this is the timeline that you can see that the basic science discovery that was in 1987 was turned to a technology by now in after 20 years. So someone has to break the ice. Someone has to have that spark, that a science has to be turned to technology. Otherwise, the science will always remain in papers. It will not come with technology if some engineering has not been done to the basic science. Okay, now here I have to take the name of few scientists who are equally important for this. So here is Professor Virginia Sixsnys, who is very important for this discovery. So he is Professor Virginia Sixsnys. So he actually discovered the same system independently in 2012, but he was unlucky because he first sent his paper to Cell. Cell rejected his paper, telling that Cell rejected this paper, telling that it is not editorial staff has rejected. After a lot of rejections, he could publish in PNAS in 2012. But by that time, Charpentier and Doudna has published the paper, their paper, independent paper in Science. So he's kind of little bit overshadowed. His work was overshadowed by the Doudna and Charpentier work. But many journals, including Science, also says that the third piece of Nobel Prize could have gone to this particular because he has also done same discovery in a different organism, of course. Then another big name in this area is Professor Zhang. Okay, you guys are listening, right? I'm speaking. Hello.
Yes.
Okay. So another professor is Professor Zhang. He is from MIT and Harvard. This is a broad institute. This is a joint collaboration of Harvard and MIT. He showed that this system can be done on human and mouse cell. Doudna and Charpentier did not do that. They showed in test tube that you can target or you can edit any DNA, but on human and mouse cell, it is a complicated system. It was done by Professor Zhang. although none of them got the Nobel Prize, they own several awards together. So one of the prestigious prizes is Kavli Prize in nanoscience. It was done by Norwegian Academy of Sciences. They own Sixsnys, and these two awardees got together. Professor Zhang and Charpentier and Doudna got many awards actually. Tang Prize, Philadelphia Science Prize, Gabier. These are also given in the life sciences. A lot of discoveries that are done in this kind of sciences. Okay. Now at this point, I have shown you the basic of this technique, but it will be very exciting to see a video that depicts this pathway in clarity. Although this video is designed by Professor Zhang's group at MIT, and although the sound does not transmit, so I have to perform a different kind of operation to do that. So here is the video. Please tell me, can you see the video? But the sound is not coming.
Yeah, we can see the video. Sound, no sound.
Okay, so I will run the video simultaneously in another device, and you'll hear the sound together. Can you hear the sound now?
Yes, we can.
Please look this video.
Every cell in our body contains a copy of our genome, over 20,000 genes, 3 billion letters of DNA. DNA consists of 2 strands twisted into a double helix and held together by a simple pairing rule. A pairs with T and G pairs with C. Our genes shape who we are as individuals and as a species. Genes also have profound effects on health, and thanks to advances in DNA sequencing, researchers have identified thousands of genes that affect our risk of disease. To understand how genes work, researchers need ways to control them. Changing genes in living cells is not easy, but recently a new method has been developed that promises to dramatically improve our ability to edit the DNA of any species, including humans. The CRISPR method is based on a natural system used by bacteria to protect themselves from infection by viruses. When the bacterium detects the presence of virus DNA, it produces 2 types of short RNA, one of which contains a sequence that matches that of the invading virus. These 2 RNAs form a complex with a protein called Cas9. Cas9 is a nuclease, a type of enzyme that can cut DNA. When the matching sequence, known as a guide RNA, finds its target within the viral genome, the Cas9 cuts the target DNA, disabling the virus. Over the past few years, researchers studying the system realized that it could be engineered to cut not just viral DNA, but any DNA sequence at a precisely chosen location by changing the guide RNA to match the target. And this can be done not just in a test tube, but also within the nucleus of a living cell. Once inside the nucleus, the resulting complex will lock onto a short sequence known as the PAN. The Cas9 will unzip the DNA and match it to its target RNA. If the match is complete, the Cas9 will use 2 tiny molecular scissors to cut the DNA. When this happens, the cell tries to repair the cut, but the repair process is error-prone, leading to mutations that can disable the gene, allowing researchers to understand its function. These mutations are random, but sometimes researchers need to be more precise, for example, by replacing a mutant gene with a healthy copy. This can be done by adding another piece of DNA that carries the desired sequence. Once the CRISPR system has made a cut, this DNA template can pair up with the cut ends, recombining and replacing the original sequence with the new version. All this can be done in cultured cells, including stem cells that can give rise to many different cell types. It can also be done in a fertilized egg, allowing the creation of transgenic animals with targeted mutations. And unlike previous methods, CRISPR can be used to target many genes at once, a big advantage for studying complex human diseases that are caused not by a single mutation, but by many genes acting together. These methods are being improved rapidly and will have many applications in basic research, in drug development, in agriculture, and perhaps eventually for treating human patients with genetic disease.
Okay, is the presentation back online?
Yes.
Okay.
Yes, yes.
So hope you have enjoyed this video that depicts this CRISPR-Cas9 cutting process and how it can do genome editing and what are the applications involved. Now, as I told you, the genome editing is not new. It has been done by people by using other systems, but the CRISPR-Cas9 has taken to a different level. So this is the triumvirate, or the three corners of the genome editing process. One is CRISPR-Cas9 on the top, you can see, and then the other one is the telane, or it is known as transcriptional activator like effector nuclease. So it is kind of a little bit complicated. So until this transcriptional activator like effector, until this term is a protein and this nuclease is another protein. So it is a protein combined with a protein that can target DNA. But the difference with CRISPR is that it uses RNA to target DNA. So that is the difference. Similarly, the zinc finger nuclease is also, zinc fingers are also proteins which are given by zinc, which are actually in the active site there is a zinc ion, and then it can be attached to a nuclease to cut the DNA. So the difference between these techniques are that the first two techniques, the zinc finger and telane, okay, they are protein based. So protein based in the sense they use a protein to target the DNA. So but the problem is that proteins are huge molecules. It is difficult to synthesize the designer proteins to target your DNA. So that is the problem with this both of these techniques here. The zinc fingers binding to the DNA and the nuclease will cut the DNA. Nuclease could be like Cas9, the different kind of nuclease people use here, but it's kind of similar function as Cas9. His function is to do cutting the DNA. The Cas9 will do the cut. It is the recognition that is important. So here the recognition is provided by the protein in these two methods, but the CRISPR uses the RNA. I told you it is the guide RNA, which is a short nucleotide. It is easy to synthesize in lab, or you can use machines to synthesize, or you can use the commercial, there are enterprises who synthesize this guide RNA. Because these are short and these can be synthesized easily, and then you just mix with the Cas9 protein and that has the complementary sequence with the foreign DNA, it will make the cut. So this is the RNA based, and it is fast, it is precise, it is cost effective, and it is scalable, because it is difficult to put proteins inside the cell, which are exogenous proteins, but these are short RNAs can be easily put inside the cell. So this customizability and the wide applicability of CRISPR has taken the genome editing to a new level. But all of them will use a nuclease, because that is the protein that will cut the DNA. The phosphodiester bond that I told you is being disrupted in that process. Okay, why CRISPR is so popular? Just I told you, it is the speed and the price that is work, the simplicity of the system. You need the Cas9 protein; it can be purified easily. You need the guide RNA. You need to make the guide RNA. You know the sequence. It is the sequence. It has to match to the target DNA. So the sequence matching is ATGC only. A will pair with T, Z will pair with C. That is the only base pairing rule in DNA. Then it is the accuracy and the specificity of the system that has made this system very popular. Okay, you can I can give you an example here. To generate a transgenic mice or a mutant mice, it takes about 6 months. Using other techniques, it takes about a month. Still a lot of time, but as compared to other techniques, it is very short. Okay, you can see the CRISPR publications or the research in CRISPR, the funding in CRISPR has gone to exponential growth, and others are usually in the baseline only. Okay, but this system is still not the perfect one, because it is only there for just 8 years. So scientists are still trying to find different ways to optimize this system and make this system even better. So some of the limitations are the limited PAM sequences. I told you that it needs a 3 nucleotide sequence, MGZ. That PAM sequence may not be available in many DNA that you want to target. Then if you silence a gene, it may have off-target side effects. So this is critical because it may lead to other things like cancer or any other diseases. Then delivery pathways. So there are people who use viruses and nanoparticles these days to deliver the CRISPR-Cas9 system to eukaryotic cells, or like difficult cell lines like mouse cells or human cells, embryos, and those are currently being developed. And the immune response, since this is a foreign system, the body immune response may also generate some responses to this technique. So people are trying to avoid and modify and rectify these limitations and make this system perfect for this kind of editing technique. Okay, now going to the applications. The CRISPR applications are manifold. Okay, so it is mainly developed for use for drug development. Okay, then to use or to develop animal models for disease disease models for testing human diseases. It is also used for stem cells. Stem cells are kind of cells that can lead to all kind of cells. It's kind of a pluripotent stem cells. People are also using now in materials. They are using in gels, hydrogels. People are using CRISPR to monitor hydrogels. Then in food, people are using genetically modified crops and other things to increase the nutritious value and the drug resistance, pest resistance of the food. Then cleaner fuel, you can make biofuel from algae using CRISPR-Cas9. And perhaps the most important is the gene surgery or genetic therapy that you can cure genetic diseases. Okay, I'll now take the example of few genetic diseases that people are using. One is in medical sciences, it could be a game changer. So in cancer, people are for example, in China and in USA, people are trying mostly in esophageal cancer. The clinical trial is going on to disable a gene here. This is the PD-1 gene. So PD-1 gene, what does it do? It tells our CRISPR system not to attack the immune system. So PD-1, people want using CRISPR, it disable the PD-1 gene, then it will tell our immune system to target the tumor cells. So it will kill the tumor cells by using own body immune system. So this is going on in China and USA. And HIV/AIDS, you all know this is a dreadful disease, because viral disease that weakens our immune system. So people are trying to cut the DNA of the latent HIV virus. Latent meaning that is silent, but it can attack in the later. It is a carrier, people with carrier virus. Then to disable a gene that is known as CCR5. This gene actually helps, that gives immunity. Mutation to this gene gives immunity to HIV. People become immune. Some people are naturally immune because of this mutation in this gene. Scientists in Japan are trying to do that, to mutate this gene to make people HIV resistant. Okay, would not be that wonderful? Okay, then hereditary blindness. So there are some diseases that because of some problems in the genetic disorders, the retina of the eye is not receptive to the light. So that can be corrected by CRISPR. In fact, it was published recently in Nature that this is the first CRISPR therapy to be administered in the retina of the eye. So it was administered in the retina of the eye to correct the gene known as CEP290. That gene mutation, the mutation in correction of the mutant CEP290 gene will give you receptivity towards light and will cure the blindness. Then blood disorders. So sickle cell anemia and beta thalassemia are the two disorders that is important for hemoglobin, or those use the hemoglobin in those diseases. The hemoglobin is in the blood is impaired, and the oxygen carrying capacity of the blood is gone. So people are trying in Europe and Asia, Europe and USA. People are trying to disrupt those mutations and produce fetal hemoglobin that is useful. Okay, in fact, in ICIB in New Delhi recently, a PNAS paper was published. They also use some CRISPR-Cas9 method to treat sickle cell anemia. So in India also, people are doing CRISPR-Cas9 regularly in some of the established labs. Okay, cystic fibrosis, one particular disease. It is a lung disease because of the defect in the CFTR gene, which affects the iron and water transport. Okay, that leads to mucus and it blocks the airways. This is a dreadful disease. People difficulty breathing and finally it leads to death. So people are trying to cure that gene to create to cure cystic fibrosis, which is a lung disease. Okay, then muscle disease, muscle disorders are also being taken care of by this kind of CRISPR-Cas9 method. So the gene responsible for causing muscle dystrophy or muscle degeneration is being taken care. Now, another important is the neurological disorders. So neurological disorders are one that has to do with the nerve cells in the brain. So Huntington disease, Alzheimer's, autism, these are some of the neurological disorders because of genetic defects. So CRISPR-Cas9 people are trying to use this technology to correct the gene for neurological disorder. In fact, Professor Zhang at MIT is specialist in neurological disorders. Okay, now in agriculture, I'll just give you 2-3 examples. You can use genetically modified crops to enhance the yield and drought resistance, as well as the pest resistance and the nutritional values. Genetically modified animals, people have tried in pigs, cattle, horses, chickens. Chickens are made disease resistant. So people are going to a next level of genetic engineering. Okay, in fact, genetically modified, CRISPR modified pigs are being explored for organ transplant. So pig kidney and liver are very similar to human kidney and liver. So people are trying to modify those genes which are will be similar to human and can be used. Pigs can be used for this kind of transplantation, because this is kind of organ transplantation is a big medical thing that is required. Okay, then greener fuels, as I told you, you can engineer crops like a jatropha as well as algae to make greener fuels for biofuels. Okay, perhaps the most exciting and ambitious project will be the ecosystem. You can engineer the ecosystem. How can you do that? You can eradicate the pest. You can eradicate some disease causing or malaria causing Anopheles mosquito. So these trials are going on in Imperial College London in UK. Okay, they have recently published in Nature Biotechnology that they have tried to make the Anopheles mosquito. We all know that the female Anopheles mosquito causes malaria. So what they are trying to do gene disruption in Anopheles mosquito to make them infertile. They cannot breed anymore. So the entire progeny and the entire progeny will be destroyed. So they have seen that after 7 generations of the gene drive technology, that means the insertion of the CRISPR-Cas9 gene cassette, they are becoming infertile. They cannot produce eggs and they cannot carry malaria. They are even trying the Plasmodium falciparum. This is the organism that causes malaria, trying to manipulate the virulent or the disease-causing genes in those organisms to make it malaria free. So you can imagine a world in future that will be malaria free.
Can I ask a question, Srinath?
Yes.
Hi Nilkamal. This is Deepak. I'm in the physics department.
Yes.
So apologies for breaking protocol, but I'm not waiting till the end. So my question is that, see, you're very optimistic about this technology, and there is good reason to be optimistic.
Yes.
But I mean, shouldn't there be some sort of regulatory mechanism in place? Because you see something like this, where we are trying to make some organism infertile.
Yes.
Tomorrow that particular genetic sequence could end up in us, affecting us in some way, right?
Okay.
It could lead to the infertility, maybe not of humans, but of some other organisms other than mosquitoes, right?
Yes.
Let's say honeybees.
Yes.
Let's say honeybees. And if honeybees become infertile, then human civilization is gone, right? Because we depend on honeybees for our food.
Yes, very nice question. Actually, my next slide will be on that only, regulation and all these things. So first thing is that the genes do not match. So it is not like that. This is designed to target the genome of Anopheles mosquito. Those gene sequence, the guide RNA is prepared like that. So
that is fine. But once things are let out into the wild,
Yes, 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 yes.
Nature takes
So
over.
yes, yes, you are absolutely correct. There is regulation that is going on, and all these things are tested in lab only. Okay, so these are now not into the wild. So these are some of the power of the technique that people are trying to evolve, and they're trying to see if you can wipe out some of this disease causing. Then the ecological imbalance that it is going to create, that is a big hue and cry about that. So that I'm going to talk to you about that in the next slide, where people have tried they modified the human genome and made people HIV resistant. So I'm coming to that in the next slide. Yes, your concern is absolutely correct that this is going to create imbalance in our ecosystem. So it is kind of a synthetic ecosystem that you are making, but we have to strike a balance between the two. Okay, technology as well as the ecosystem. So what you said is absolutely correct, that ecosystem is going to be hampered, and it can be disastrous to humans as well in the later future. But I'm coming to that particular slide in the next one. Okay, thank you for asking that question. The next is the de-extinction. So people are trying to do something that is unthinkable. So it is like a science fiction coming alive. You have probably seen the Jurassic Park, where the dinosaurs was a velociraptor was taken alive from DNA sample, but this is actually doable using by this CRISPR-Cas method. So people are trying to do woolly mammoths. So they are actually targeting those ones which are not harmful. Okay, passenger pigeon and Tasmanian tiger, which are extinct. So people are trying to bring back those kind of science to this on the plate using this technology. So let me go through the woolly mammoth. They were extinct, and they were present in Siberia. So they were present in Siberia, but because of ecological imbalance, climate change, they got extinct. The closest relative is the Asiatic elephant. So the genes of elephant are almost 99% similar to this Asiatic elephant. Only they have extra genes for like fur and survive the cold. All those kind of additional genes are being tested. And Professor George Charles at Harvard Medical School is leading this effort of resurrection or restoring of this animal which once walked on the walk on the earth, which is a very good good for the environment. Okay, so they have also tried passenger pigeon. So this is a pigeon that was once abundant in North America. People have tried. The closest relative is the bentail, because the genomes match to 99% almost. But some genes which are from the passenger pigeon is now in the lab. Obviously, this is nothing is in the wild. All in the trials only in the academic labs as well as the other things. And these are the two scientists, Professor Ben Novak and Beth Shapiro. Those are in the UC Santa Cruz. They're spearheading this method. Okay, then Tasmanian tiger. This was the tiger or tiger look-alike that is once very popular in Australia, in Tasmania, in Australia. It has gone extinct. Now scientists are trying to, Professor Andrew Pask at Melbourne University is trying to resurrect or restore this life. So all these initiatives are funded by a non-profit organization called Restore and Relieve and Restore, who are targeting this kind of extinct organisms. Okay, who who is once now extinct, now becoming a probability in the future. Okay, although this is all good about science and technology, there is a lot of business also in this method, in this process. So there is a huge patent battle going on between Feng Zhang, because people have realized this is very promising technique. So both between Harvard and MIT and California Berkeley, legal battle is going on to acquire the patent for some cases for CRISPR-Cas9. So this is in court currently, but this has not stopped people from trying on all different editing techniques. Okay, lot of businesses grown or companies have formed using CRISPR-Cas9. Among them, the Editas Medicine, which is the main stakeholder, is Professor Zhang of MIT. Okay, they are using for different diseases and crop modeling. Okay, Intellia Therapeutics, which is Professor Doudna, a main stakeholder. It is based in California. Okay, they are using CRISPR-Cas9 to do mainly for diseases, genetic diseases, and crops and livestock. Okay, CRISPR Therapeutics is based in Europe. This is Professor Emmanuel Charpentier. And these are the four scientists that are very much prominent in this entrepreneurship of the CRISPR-Cas9 strategy. So a basic science discovery turned to a technology, now becoming a huge market for therapeutics and drugs. Okay, Now, the question that the debate, that bioethical debate that Professor Deepak just now pointed out, that it has to be regulated. In fact, a thing that has happened in China. So because of less regulations or some other uh, uh, absence of ethics regulations, one professor, Zheng Kui, in Southern University of Science and Technology, he has edited the embryos of humans, okay, and then in vitro fertilized to make it to HIV resistant babies. So there were Lulu and Nana. Those were known as CRISPR babies. Initially, they were appreciated, but because of lack of ethics behavior, lack of permission to do this kind of research, because you can stop at the embryo stage, but you cannot make a human out of that or living system out of that, because someone is going to inherit the gene, will pass on from one generation to another. So this is going to be very critical, and scientists have come up with this option that more research need to be done before this application approach need to be done on humans. So in fact, this professor was later on was jailed, and because of breaking the ethics of research. In fact, scientists are coming up with lot of talks and seminars telling that a moratorium on human germline editing has to be stopped, because there should be a ban, because there should be a moratorium for not applying to human embryos. Embryo stage is still fine, but it cannot be made to make babies or CRISPR babies. Okay, so in fact, Jennifer Doudna, who is the creator of this technique, she anticipated this problem. She wrote a book in 2017. I urge the public. This is a general science book, not a lot of technical things, but this talks about the ethics, science, and the power of this technique. She told that a crack in the creation. So although this technique is very powerful, there is still a harm. There is a looming danger that unscrupulous people can use it for bad purposes. So she already anticipated this problem, and she is one of the great propagators of this technique that moratorium has to be there. And all the scientists, recently a article came up in Nature that including Feng Zhang and Emmanuel Charpenter, all the big guys of the CRISPR-Cas9, they all came up with this with ideas that there should be a ban, there should be a moratorium for human editing. So this technique is powerful. This technique is very useful, but it has to be controlled and has to be regulated. So all the countries are encouraged to form regulatory strategies for this. Okay, so with that, I'll come to the conclusion that this is a technique, this is a basic science that has been turned to a powerful technique where guide RNA targeted genome editing, okay, including the Cas9 protein. It has a lot of application, as I told you. This is a wide application in medicine, agriculture, and biotechnology. This uses the ancient defense system of bacteria, but a lot of it has to be controlled by regulatory strategies for editing human genes, and also disturbing the ecological behavior, okay, of different techniques that is in the future. So within just 8 years, this technology has reached this level. Okay, with that, I would like to I hope that I have given a glimpse of this CRISPR-Cas9 from discovery to the Nobel Prize to the current status. And I thank the Department of Physics for giving me, NIT Surathkal for giving me the opportunity to present in this colloquium. And I also thank the audience for listening to this talk. And I'll be happy to take any questions. A little disclaimer, I'm not a biologist and neither I use this technique, but I'll try to answer as many questions as I can. Thank you.
Thank you, Dr. Nilkamal. It was a wonderful talk. And there are a lot of questions indeed. So I will read one by one and maybe you can answer. Okay. So Professor Satyanarayanan wants to know, how do I experimentally determine the cutting of DNA and addition of a new chain?
Hello, you can sequence. So you can, after cutting the DNA, the size of the DNA reduces. People uses one electrophoresis, which is known as agarose gel electrophoresis. You can see the sizes of the DNA because that will be different. And the addition of the gene will also, there will be size difference. And obviously the main thing is sequencing. You can sequence the gene the genome or whatever you are targeting, and it will give you the exact sequence, whether your sequence has been incorporated or it is not. So it is mainly, it is known as Sanger sequencing. So Frederick Sanger was the person who discovered the DNA sequencing, also got the Nobel Prize. Okay.
Upas Yeshraj, he asked, how do we map the gene sequence to a particular disease? What is the how do we identify a certain gene sequence to a disease?
Yes, that is a very interesting question, and actually there are a lot of genetic diseases people do not know the responsible genes. Okay, it is a very complicated system, especially in humans, because the genome is so big. So let me give you a simple example. The sickle cell anemia. Sickle cell anemia is because of a defunct form of hemoglobin. Okay, in that hemoglobin, the protein, one amino acid, the valine amino is, or I'd say glutamate is an amino acid. It is converted to valine. Just one change. One change makes the hemoglobin protein not to fold properly, and it becomes a sickle step. The original step is lost. So it is very difficult actually. But scientists, coming back to your question, people do RNA sequencing, all sort of sequencings, and they usually use genetic engineering. They mute silences in, and they see the effect. Okay, so uh, genetic engineering is actually used to know the cause for any genetic diseases. You have to mutate the gene, and you have to see the effect. So it is done at DNA level, RNA level, as well as the protein level. So a lot of research has to be done in this case. Okay.
Dr. Shahjahan has a related question. So, to cure a disease in a living organism, should we target the entire organism, or targeting a one particular gene is sufficient?
One gene, even the little bit mutation. I told you, in hemoglobin, one amino acid change will give rise to sickle cell anemia. That anemia. So you can yes. Yes.
Can I ask a question? If it's a living organism, there are a lot of cells and the same gene will have several copies. So if you are using genetic engineering to cure a living organism, do you have to target all the cells in the body, or a certain subset of the cells are enough? That's my question.
Um, I think people target the bone marrow also, where the birthplace of most of the cells. So bone marrow is targeted for in many cases, where it will give rise to cells, and it will go to all different organs. Okay. Um, um, exactly, I'm not sure at this point, but um, it will be difficult, because these every organ have different cell types, and it is difficult to target all of them at once. So probably the bone marrow cells will be targeted, which will be later on passed on. The proper copies will be passed on to all the organs. So that people also do in pluripotent stem cells. Stem cells can give rise to all different cell types. So if you change the gene in the stem cells, then all the change is carried toward all different cells.
Yeah, but that can be done only in the embryonic stage, right?
Embryonic stage. In living organisms, it is complicated. It is complicated. I feel in where the sickle cell, the clinical trial going on in Europe and Asia to target the blood disorders, sickle cell anemia, they are targeting the bone marrow to cure the copy of the gene that codes for hemoglobin, and that will be transferred to all the other cell types eventually.
So Anandram Anandram has a question. So how efficient is the scaling up process, and how is it usually done of CRISPR-Cas9?
Scaling up in the sense you can target many genes at one time. So that was also shown in the video, because the thing is that you have to I have to give the difference between the other genome editing techniques, the zinc finger nuclease, as well as the TALEN. So there you have to synthesize the protein to target the DNA. So to synthesize the protein, it is difficult because protein is a big macromolecule. You have to take care of the folding and all those things also. Here, you can design the RNA, the guide RNA, which is a short nucleotide, and you can target many genes at once. So you can do simultaneously genome editing of many genes at once. So that is the scaling up that I was talking about. So you can design many RNAs. All of them will be bound or partnered with Cas9. They will make the genetic change at the same time simultaneously.
So there is a question by Naveen Kumar on de-extinction. So can this be used to learn prehistoric human migration? Something related to the work done by David Reich and team.
Wow. It is a wonderful question. I don't have the answer to that. For example, the de-extinction project, that is kind of a the most ambitious project so far on CRISPR, and this migration of people, the demography change, and all those things. Probably yes. I don't have an answer to that at this point, but you probably have to have the genetic samples. Some footprint of those genetic samples has to be collected to study that. I think. That's a wonderful question.
So if there are any further questions that someone would like to ask, now is the opportunity. They can unmute themselves and speak.
Yes, please. I'm also a novice. I am in this technique. I just knew about this technique, but the power and the adaptability of this technique is enormous.
But that was a great talk, Nilkamal. It was very interesting, very engaging till the end.
Thank you.
Thanks.
Thank you. So, any questions from any students, maybe?
Yeah, Professor Nilkamal, just one comment.
Please.
It was so wonderful talk, and thank you very much.
Thank you.
And just one comment. I think the greatest engineer is the nature, and it's so complicated. The way you are talking this editing, I think human being has actually recognized that there is a mechanism, right? It is not that they learn from nature and then are and using it.
Yes. They learn from nature. Yes.
They learn from nature. So fundamentally, nature is so beautiful, and it's wonderful engineer itself.
Yes.
I just want to.
Yes. Very nice comment, because I wanted to extrapolate on that. I told you this entire system has been taken from bacteria. Okay, so how bacteria kill the virus? That is the basic science. How bacteria kill the virus. So that was engineered by the Professor Doudna and Charpentier. That is another thing. But now bacteria has designed this strategy to kill the virus. They have not attempted for editing or anything. Their only goal is to kill the virus, to survive, to make them survive. Okay, but now viruses came to know that the bacteria has this CRISPR strategy. So they have designed another strategy to tackle the CRISPR. That is another beautiful story that was published in Nature, that the virus are even smarter. Now bacteria has to play one more dice. I mean, it is kind of remarkable how these little things play each other. And one more thing I need to tell, that this CRISPR strategy is also applied for COVID-19. So people are trying to cut the viral DNA of SARS virus, SARS-CoV-2. In India also, people try. Tata Chemicals is doing, and with collaboration with IGIB, and some other people are also doing in the world, targeting the SARS virus, COVID virus, using CRISPR to cut the DNA. Because once you can do that very well, then you can disable the virus, and you don't need a vaccine for that, actually.
So you think there is a silver lining for COVID-19?
I mean, people are trying. It's in clinical trial. I think before that, the vaccine may come. The vaccine itself is a virus. I told you, vaccine is no drug. It's a virus to train your immune system. Okay, but if this is kind of a foreign material like the CRISPR-Cas9, but the problem with the virus is this kind of, even the HIV virus, they mutate very fast. Why there is no vaccine for HIV so far? Because HIV virus mutates its DNA so rapidly that this kind of strategy has less time to adopt, kind of. So similarly with COVID as well. So mutating viruses are difficult to target. It takes about 10 years to develop a vaccine. Some of the successful vaccines will be like hepatitis B, polio, those kind of, because those viruses are somewhat less mutating as compared to HIV and COVID.
Okay. So, Nilkamal, one small doubt.
Yes, please.
You are talking about COVID-19 mutation. So, what is your hope that this mutating to a lesser, dangerous species, or can be very dangerous one also? What?
It is a flip of a coin. I mean, it can be both ways.
Okay. So there is no rule that it will be going to be like common cold after some time. It's such a way that it won't be
less dangerous.
Yeah.
I mean, that is also, I mean, nature strategy. Human has no control on that, I think. So they will adapt. So I mean, I don't know it is true or not. It is being said that the COVID-19 virus strain that is in India, that is giving the pandemic in India, it is different from China. It is less virulent than China. That is kind of a mutated virus. But still, it has the property to cause the disease, but it is not as dangerous as the original Chinese virus. But when it comes to India, when it comes in someone in carrier, somebody else's people's as a carrier, it may mutate when it goes to another person. So all those things can happen. But whether it will become common cold in near future, it is difficult to say, sir.
Yeah.
But it has very similarity to SARS virus that was quoted earlier.
Thank you.
Professor
Thank you.
Nilkamal. Professor
Yes.
Nilkamal.
Yes, please. Yes, please.
Good afternoon, Nagraj here. I just missed, I think, whether you have addressed this question previously. Just wanted to know what kind of lab infrastructure you need to handle this. For example, scissoring the RNA, or what, where this what is that virus, CRISPR virus,
Yes,
right?
yes. Actually,
Where
not
is it available? Is it something commercially available, or
no,
and
no.
how do you experimentally, how do you go about it,
Very good.
introducing that, handling that, and checking the effect of that and all those things?
Very good. Very good question. Let me. Can you hear now? Hello, can you hear now?
Yeah.
Yeah, you are audible.
My voice
I
was
think Nagraj sir wants to know whether we can afford it in an Indian setting or not.
Oh yes. Many labs have started doing. It is not very costly. I told you, it is the biggest advantage of CRISPR is cost effectivity. So you need the protein. The protein you can make in the lab. So protein that comes, this particular system protein comes from the organism known as Streptococcus pyogenes. That is the bacteria that causes fever. But people have extracted the gene that is used to make the protein. We can make protein in the lab. We make in my lab here, we make protein all the time. So you can express the protein in a strain like E. coli, which is a lab organism, and you can purify the protein Cas9. So one part you have very cheap. The Cas9 protein you can make in the lab itself. Then the second part is the guide RNA. The guide RNA synthesis, you need a specialized setup. So there is a nucleotide or DNA oligonucleotide or DNA synthesis machines are there. You can either, if you have that machine in the lab, the machine is not that costly. So if you have that in the lab, you can do that. Or there are a lot of companies who synthesize these guide RNA. So you can, based on the sequence, you give the sequence. Okay, it would have been better. It would have been better if I show the sequences of this. It is simple ATGC pairs. Okay, so if you give the sequence, it will synthesize that piece of DNA or RNA for you. Okay, then the target DNA where you want to do. Okay, now it is easier. This technique easier to do in a prokaryotic cell. Prokaryotic matlab single cell, like bacteria, fungus, those ones. But if you go to eukaryotic, which are bigger ones like mouse cells, human cells, or even yeast, it is difficult, because then you have to have specialized technique for putting the plasmid inside the cell. So I have that slide actually. Um, can you see the slide or no?
Yeah. Yeah.
So this is the slide where the guide RNA, the protein, you can put in a plasmid and you can put inside the cell. Okay, so the protein is.
These are handled manually with scissor and all, or do you need some
No
advanced
Caesar.
microscope and all?
Caesar here is like protein amino acid cutting DNA strand. There is no Caesar like manual scissors. Okay, it is just a analogy. It's just an analogy.
Nilkamal,
So
one second.
yes.
There is an echo coming from someone. Nalasa, maybe you can mute your mic.
Yeah. Now it is fine.
Now it is fine.
Yeah, that probably because you logged in two device and it is.
Who,
Yeah,
me?
that may be the only reason. Yeah.
No, now it is fine. Now it's fine.
Now it's fine.
No, it is fine. Yeah. So what I'm telling that it is the protein that cuts the DNA, and the protein, there are some residues. Actually, it activates water, and water will hydrolyze the phosphodiester bond in terms of chemistry. Okay, that has been given the analogy that it cuts like a scissor. When you cut with a scissor, it makes blunt ends. Okay, like similarly, it will also make like kind of blunt end. It will cut the both ends, both strands of DNA together. So it will make a blunt end and separate the pieces of DNA into two different pieces. So it is kind of the protein that cut the DNA. So that is why it is just given an analogy with a scissor. No manual scissor is required for this process. So it will do inside the cell. The protein will cut. After that, you run the gel, you will see that your piece of DNA is half the size because it is now separated into two pieces. So people can use agarose gel electrophoresis for this. And some other techniques, of course, sequencing can do. So it is not so. Coming to your original question, can you please mute, sir? Nagraj sir. Coming to your original question, it is not costly at all. So you can make the protein in your lab. You can give someone the DNA RNA sequence to make. Then you can put inside the cell in your lab. It is not cost effective at all. People are doing regularly in India. I have given a few examples. IGIB in India and CBS is also doing. I think ISC also some labs people do. In fact, in the BSB lab, biosciences department here, Professor Sudhanshu Sukla's lab in IIT Dharwad, they are using CRISPR-Cas9 to target some genes. So it is very common. It has become very common. That's why the popularity and the usability, applicability of the technique.
Okay.
I think Shubham Gupta wants to ask.
Thank you very much.
Yeah, thank you so much. Thank you so much.
Shall we go to the question, and then we will wind up?
Yeah.
Why the coronavirus do not spread in cows, dogs, but in human? I don't know. Maybe these cows and dogs have some, just like bacteria has a strategy to kill the virus. These are animals also may have that strategy to some kind of weapon to kill the virus. So the best way is to, in a scientific way, to compare the genomes of a cow and a human. Probably you can see what genes are there in the cow which are not there in the human. Okay, and then you can see those genes.
Nilkamal, I think in South Korea there is a cat. Cat was infected by COVID-19, and I think US also there is a tiger that was infected by COVID-19. That's what it came in the news, but I don't know exactly what happened thereafter. It's not that it's only humans are getting infected. I think it could also be animals, as per the news, but there is no scientific evidence.
Yes, so there is a lot of these research need to be done, but exactly at this point I don't know why it does not spread in cows and dogs. It has to be do with the genetic makeup. The genetic makeup has something to do to tackle the virus.
Yeah,
but good question indeed.
So
Srinath,
thank you very much.
yeah.
Thank you very
Any
much,
other
Dr.
question?
Nilkamal. It was a very interesting talk.
Thank you.
Very
Thank you.
engaging. You gave a very whole picture of the
Yes.
this year's
I did not cover a lot of, there are more science into it, but I thought it is a general audience, and I kept the biology to minimal and try to make as general as possible. But there is more to it. It's more complicated than what I have presented today.
Yeah. Thank you very much for your time.
Thank you.
And thank you all the audience.
Yeah.
Thank you very much.
Thank you. Thank you,
Yeah,
Karan sir.
thank you. Thank you, Nilkamal Mahanta. It was very interesting, very nice. Definitely, after COVID, we'll be inviting you into department for
Sure.
a
Thank you.
much wider audience. And it was
Sure.
very nice. Thank you so much for your time. And
Yes. Thank you for inviting and giving me this opportunity and for this talk. Okay. And I'll join again on 10th for the other talk, because those talks are also not from my field, but those are coming from experts. So we always it always gives some spark when these experts talk. Okay. I'm a novice in this field, so I've just started my career 1 year back. Okay. Thank you.
Thank you very much.
Thank you so much. Bye.
Bye.